Before we begin, we are going to introduce the models used in our representation of DNA replication. Today we will be showing you how DNA replicates.
DNA replication starts with the enzyme helicase unzipping the DNA by breaking the hydrogen bonds between the bases. This will form a replication fork.
First, we will focus on the leading strand. RNA primase attaches a primer at the 5' end of the strand.
Next, DNA polymerase III builds a new strand of DNA by adding complementary nucleotides. Starting at the primer, DNA is built by adding to the 3' end.
Meanwhile, the lagging strand is also being built. RNA primase attaches a primer and DNA polymerase III adds nucleotides to the 3' end. This newly formed piece of DNA is called an Okazaki fragment.
Another primer is placed farther down the strand and polymerase III adds more nucleotides.
This process repeats itself all the way down the lagging strand.
Next, DNA polymerase I replaces the RNA primers with DNA nucleotides on both strands.
And lastly, DNA ligase bonds the backbones of the Okazaki fragments together.
The end result is two identical copies of DNA.